Hi, everybody. Welcome back. Today we are going to continue with our discussion of the immune system. So yesterday we talked about lymphatic tissues and organs and their role that they play in our body defenses. They are going to serve as sites where white blood cells can proliferate and form like armies of white blood cells that are capable of fighting off infections. We looked at the thymus and the spleen, and we looked at Peyer's patches and tonsils, and those are all examples of lymphatic tissues and organs. Once we looked at our lymphatic system, we started looking at body defenses, so our body's ability to fight off invading pathogens. And we said that our body defenses are going to fall into kind of two main categories, right? We're going to have our innate or non-specific defenses, and this is going to be a generalized response to any type of an invading pathogen. Then we have our adaptive or specific immune system, and the adaptive or specific defenses is going to recognize the foreign invader on a molecular level, right? It's going to see it, it's going to spot it and go, oh, you are E. coli, or oh, you are salmonella, and it is going to it's going to mount a defense that is specific to that invading pathogen. So when we look at those two branches, when we look at our innate or non-specific defenses, and then we have our adaptive or specific defenses, we're going to have three different lines of defense. The first line of defense is just going to be intact skin and mucous membranes and their secretions, right? Just having a physical barrier between the external world and the internal world means that things that just land on you are just going to stay on your skin, right? They're not going to be able to burrow very deeply into your tissues and actually get you sick. They're just going to stay on the surface. Then we're going to have, excuse me, then we're going to have our second line of defense. And the second line of defense is like the second fence around the prison, right? So if you get over the first fence, if you get into the body through the skin or the mucous membranes, then we have this whole kind of cast of characters that's going to play a role in our second line of defense. This is going to include some of our immune cells. We're going to have macrophages and we're going to have NK cells. It involves inflammation. Inflammation is characterized by redness, heat, swelling, and pain. It includes fever. Fever is going to be an upward adjustment of body temperature. High fevers are bad, but moderate fevers are good because it stimulates our body's healing process, and it's going to cause our liver and our spleen to sequester two really important minerals, iron and zinc, and sequester those in the liver and in the spleen so that they're not available for bacterial replication. It's also going to include our antimicrobial proteins, things like interferon. Interferon is secreted by cells that are already infected with viruses. And once they're infected, what they do is they secrete these interferons that enter all of their neighboring cells, and that is going to stimulate the neighboring cells to start producing their own antimicrobial proteins, their own antiviral proteins that prevent the virus from being able to get inside of those non-infected cells. Then we have complement. And complement is going to be a set of about 20 different proteins that circulate in our blood in an inactive form. Under certain conditions, those proteins will become activated. It's called fixing complement. And when complement is fixed, it does a couple of different things. One of the things that complement does is it enhances inflammation. So it's going to enhance all of those aspects of inflammation, the redness, heat, swelling, and pain. But another thing that complement proteins do is they will form proteins that kind of cluster on the surface of microorganisms and make it easier for our phagocytic white blood cells to find them and to engulf them. So these are all going to be generalized responses to any type of invading pathogen. Now, our third line of defense is what we call the immune response. And immune response is actually going to have several different parameters. It's going to increase inflammatory response and all of our other non-specific defenses. So it makes our phagocytes extra aggressive. It enhances inflammation. It enhances fever. It is going to make sure that complement proteins and interferon proteins are going to be produced in a life-sustaining rate. Another thing that immune response does is it provides protection very carefully targeted against a specific antigen. And an antigen is something that will elicit an immune response. So anything that your immune system takes a look at and goes, yeah, you're not normal. Like, you're not part of this human being. You don't belong here. And our adaptive or specific defenses is going to prime our body for a very vigorous response if we have any future encounters with that same antigen. So the adaptive or specific immune system is going to be our body's third line of defense. And so it is going to recognize foreign molecules that we call antigens and acts to inactivate them or to destroy them. Now, one of the interesting things about the immune system is that the immune system has to be exposed to an antigen before it can protect the host from it. So it has to see it in order to know what to look for. So it's kind of like bloodhounds, right? If they use bloodhounds to try and track the scent of somebody, they'll give them like a t-shirt or something like that that person has worn. And they're like, okay, here, go try and find this. That's kind of the way the cells of our immune system work. We have to show them what they're looking for, and then they can go out and find it. So there are three aspects of our adaptive defenses that are going to be a bit different from our non-specific defenses. The adaptive defenses are going to be antigen specific. So that means we're going to recognize and act against a particular pathogen or foreign substance. So we're going to recognize it on a molecular level. It is systemic, which means it is not going to be restricted to the initial site of the infection. The whole entire body is going to kind of jump on board to try and fight this infection. And it has memory. And this means that the adaptive immune system has the ability to recognize and mount an even stronger attack on an antigen that it has previously seen. So it's like when you get an immunization, right? People are getting their COVID shots now. So by getting your COVID immunization, you're teaching your immune system to recognize certain parts of the virus. Then if you ever encounter the virus in your environment, then you've already got these memory cells that have been created, and they are going to jump into action, right? And they are going to fight that infection really, really quickly. So when we look at our adaptive defense system, we're going to see that it is composed of two separate but overlapping arms. We have humoral immunity, and humoral refers to your body fluids. So plasma, interstitial fluid, lymph, and it's also called antibody-mediated immunity because it is provided by antibodies that are secreted into our body fluids. So that's why they call it humoral immunity. The antibodies are going to be in our body fluids. Then we have cellular or cell-mediated immunity. And this is when the lymphocytes are directly defending the body. In our cellular or cell-mediated immunity, we're going to target the virus or parasite-infected cells. So these foreign agents have gotten into our normal body tissue cells. It also targets cancer cells, and it will also target cells that have been transplanted. So if you ever have a liver transplant or if you have a heart transplant, right, the adaptive immune system is going to go looking for those non-self tissue cells. Now, cell-mediated immunity is a little bit different, right? We're not going to have antibodies. Instead, we're going to have direct lysis. Lysis means to break. Direct lysis by the lymphocytes, or we can have indirect activity by releasing chemicals that enhance inflammation and activate other immune cells. So it's really very, it's like a dance, right? It's like the steps in a complicated dance that you have to learn. So before we get too much into our adaptive immune system, let's talk a little bit about antigens. So antigens are substances that are capable of activating our immune system and provoking an immune response. So this is going to be a foreign intruder or anything that is non-self, right? Anything that has a genetic makeup that is different than your genetic makeup. So examples of antigens are going to be virtually all foreign proteins, nucleic acids, carbohydrates, and lipids. It also includes things like pollen granules, microorganisms like bacteria, fungal spores, and virus particles. Now, of this list of antigens, foreign proteins are the strongest antigens. They're going to give us the most vigorous immune response. Now, our own cells are going to be decorated with a variety of proteins on the surface of the cell, embedded in the plasma membrane. And our immune system is trained to recognize those proteins as self. So it's kind of like when you're in the Army, you're trained to recognize your friends versus foe, right? You learn to recognize your uniform versus somebody who's in a different uniform. So a self-antigen, your own antigens, do not trigger an immune system in you, but if we transplant your tissues into somebody else, then it will trigger an immune response. And that explains why our bodies tend to reject the cells of other individuals unless extreme measures are taken in order to prevent rejection, right? You have to take, anti-rejection drugs, and you have to take steroids to suppress your immune system. So let's look at the cells that are important in our adaptive immunity. So we're going to have lymphocytes and we're going to have macrophages. Underneath lymphocytes, we're going to have two major types of lymphocytes. We have B lymphocytes, which are usually just referred to as B cells. And B cells are going to provide our humoral immunity by producing antibodies. Then we have T lymphocytes, which are also referred to as T cells. And T cells are going to provide our cell-mediated immunity. Now, macrophages are an important cell in our adaptive immune system, but they do not respond to specific antigens. Instead, they are going to play an essential role where they help to activate the lymphocytes. Now, both T cells and B cells are going to originate from our bone marrow, and they are released as immature lymphocytes. Now, whether a lymphocyte matures into a B cell or it matures into a T cell, that is going to depend on where that cell migrates in order to receive its education. And that is called immunocompetence. That is when we school the lymphocytes on these non-self antigens, on these antigens that don't actually belong to our bodies. And so once they're educated or they become immunocompetent, then they are capable of responding to an antigen by latching onto it. So T cells are going to arise from lymphocytes that originate in the bone marrow, but then they migrate to the thymus, where they undergo their maturation process. They get their education in the thymus. And only the the only mature cells that are going to leave the thymus are going to be the ones with the sharpest ability to identify foreign antigens. I read a paper once that said that it's a very expensive process because only about 2% of the lymphocytes that migrate to the thymus actually leave as mature immunocompetent T cells. 98% are not going to get it right, and they're going to be destroyed in the thymus. Only 2% receive their full education, and then they go out into the real world, which is our other lymphoid organs. Now, B cells are going to gain their immunocompetence in the bone marrow. So T cell stands for thymus dependent. It has to spend some time in the thymus. And B cells is B is for bone marrow. So they undergo their immunocompetence in the bone marrow. Now, once a lymphocyte is immunocompetent, it's spent time either in the thymus or in the bone marrow. Each individual lymphocyte can only respond to one antigen. And that's because every single receptor on its surface is identical. So one receptor, one antigen. And since every single receptor on its surface is identical, then that means it can only respond to one antigen. Now, lymphocytes become immunocompetent before they actually meet their antigens that they later attack. So if we encounter an antigen that we have been programmed through our DNA to recognize and attack, then that specific population of lymphocytes is going to be mobilized, right? So we have lots of different types of B and T cells that are wandering through our tissues, and each individual B or T cell can only respond to one antigen. And so when they meet up with their specific antigen, that's it. We're off to the races. If we never encounter the pathogen against which we are immunocompetent, the lymphocyte populations may never be activated. So there's a good chance that, a certain percentage of us are actually carrying B and T cells that make us immune to Ebola, right? Some of us will, just by sheer chance, through random mutations. Some of us are resistant to Ebola. But if we never actually encounter Ebola in our environment, then those Ebola lymphocytes will never be activated. They're there, they're hanging out just in case, but they may never become activated because you never encounter Ebola in your lifetime. So what's interesting here is that it is our genetic makeup, not the antigens, that determine which foreign substances our immune systems will be able to recognize. So my immune system might recognize a couple of different antigens that yours does not, because that ability to recognize antigens is passed on. So basically, our immune systems are like a snapshot of our entire evolutionary history of every single antigen that we've ever encountered that didn't wipe us out as a population, right? Because if it wiped out 100% of the population, then there would be nobody left to pass those gene mutations onto. So we have a mixture of mutations that our parents and their parents and their parents and their parents, just thousands of generations of the receptors that were able to protect us from invading pathogens that we encountered sometime during our evolutionary history. Now, after our B and T cells become immunocompetent, so B cells become immunocompetent in bone marrow, T cells become immunocompetent in the thymus, they leave and they migrate to the lymph nodes and the spleen and to other lymphoid organs. So the Peyer's patches and tonsils and the appendix, so they kind of like spread out and go into our lymphatic tissues where they can encounter their antigen and become functional. So lymphocytes complete their differentiation into a mature or B cell when they bind to their specific antigen. Lymphocytes, especially our T cells, they like to circulate through the body. So they'll be in the blood some of the time, they'll leave and go through the tissue spaces, then they'll enter the lymphatic system, and then they'll end up back in the blood. And so they're kind of like constantly in motion. So they're really seeking out those antigens that they are programmed to recognize. B cells tend to stay more in the lymphoid tissues. T cells are much more likely to migrate through the body than B cells are. All right, so we have these immature lymphocytes. Some of those immature lymphocytes are going to migrate to the thymus, where they are going to become immunocompetent, but they haven't actually recognized, they haven't encountered their antigen in your body. So they're said to be naive. They're immunocompetent, they've been educated, but they're naive, they've never had a real job. So just like when we all graduate from high school or we graduate from college, we have all of this book knowledge, but we haven't had like a real job. So that's what these immunocompetent but naive lymphocytes are like. Other lymphocytes are going to stay in the bone marrow, and they are going to undergo their education right there in the bone marrow. So that's going to be our B cells. Once they've been trained to recognize one specific antigen, each B cell and T cell is only going to recognize one antigen, they are allowed to leave, and they are going to seed our other lymphoid organs. So lymph nodes, tonsils, the spleen, Peyer's patches, you name it, all of those other lymphatic tissues, lymphoid tissues, that's where they're going to migrate. And they will not finish their maturation unless they actually encounter their antigen in your body. They never get the real job. They move back into the parent's basement. All right, so that's our lymphocytes. We said that macrophages play an important role in immunity, but they don't directly fight the specific antigens. The macrophages are going to be distributed all throughout our lymphoid organs and connective tissues. And they are going to arise from a specific type of white blood cell called a monocyte. And those monocytes are derived from the bone marrow, as are all of our different types of blood cells. And macrophages are capable of phagocytosis, right? They are going to engulf foreign particles, but then what they do, it's kind of a really neat trick. They capture these foreign antigens, they engulf them, they use digestive enzymes that are in the lysosomes and chop it up, and then they display a little piece of that foreign antigen to our immune cells. So they are called antigen presenting cells. They capture the antigens, chop them up with lysosomal enzymes, and take a little piece and display it on their surface, and then they go and show it to the T cells and say, is this your antigen? Is this your antigen? Is this your antigen? So they're trying to find the correct T cell for that specific antigen. Now, macrophages can also release chemicals that will activate the T cells. So not only do they have to show the antigen to the T cell, the chopped up antigen, they also release chemicals that activate the T cell. And then in turn, the T cell is so happy about it, it secretes its own chemicals that make the macrophage even more aggressively phagocytic, right? So they're just going to like turn on the turbo and go out there and they're going to fight the infection. Now, macrophages usually stay in the lymphoid organs. So spleen, not the thymus, the spleen, the Peyer's patches, appendix, the tonsils, and so forth. Now let's look at what happens with the B cells. So this is our humoral or antibody-mediated response. So when you have an immunocompetent B cell, it's undergone its education in the bone marrow, it leaves the bone marrow, and it's usually going to go hang out in a lymphoid tissue like a lymph node, right? It's going to go hang out in a lymph node. Once it's immunocompetent, it will not be stimulated to complete its development unless its proper antigen binds to a receptor on its surface. So the binding event, when the antigen binds to the receptor, that is going to turn on a switch inside of the lymphocyte, and that specific lymphocyte that has the receptor for that antigen, it is going to undergo a process called clonal selection. And all that means is that specific B cell is going to divide and divide and divide and divide and divide and divide and form an army of cells that all have the exact same receptor as that first cell that was activated, right? So we have this army of clones that is capable of responding to the antigen that set this entire process off. So each one of those daughter cells is a clone of the original B cell that was activated. Now the clones are going to give us our primary humoral response. So most of our clones are going to become plasma cells. And plasma cells are going to be the antibody producing cells. They're going to churn out antibodies at the rate of about 2,000 antibodies per second. So, I mean, when I tell you it kicks it into turbo gear, it really does. So they just start kicking out these antibodies, and then the antibodies float around in the plasma and they clump on to things that don't belong. But some of the clones are going to become memory B cells. And so they do not become plasma cells, but they remain behind in our lymphoid organs in case we ever encounter that same antigen again. It's like we've trained our immune systems to go, oh, I know you. You don't belong here. And so we have this army of clones that's capable of responding to that same antigen. And we have lots of memory cells that are created. So if you ever encounter that antigen a second time, your response is going to be faster, it's going to be more aggressive, and you're going to eliminate that antigen from your body much more quickly. That's called the secondary response. So anytime you encounter an antigen after that initial or primary response, the immune response is going to be faster, more prolonged, more effective, because all of the prep work for the attack has already been done. We've created all of these memory cells. All right, so let's see. This is 3 different types of B cells. Now, B cells are different from T cells because B cells are going to respond to antigens in our body fluids, right? They're just going to be like drifting by, and the B cell goes, oh, look, yeah, it's like when a cockroach flies by and you're like, oh crap, that doesn't belong here. So it goes by and it's like, oh, yep, that doesn't belong, and it goes and gets it, right? So we have 3 different types of B cells. One B cell has green receptors, so it's a receptor for one type of antigen. One B cell has dark blue receptors. That's going to be a receptor for a different antigen. And one B cell has light blue receptors, and that's going to be a third different antigen. So we have hundreds of thousands of these circulating in our bodies, right? And they all have different types of receptors. But each individual B cell can only respond to one antigen because every receptor on its surface is molecularly the same. So this B cell receptor is going to be identical to this B cell receptor, which is identical to this one. So this B cell can only respond to one antigen. So in this case, we have this B cell that has the dark blue receptors. And so we just happen to get the correct type of antigen. That antigen is going to bind to the B cell receptor. And so that kind of completes the process that is required for that B cell to finish its maturation. So this B cell, it now has its antigen that's bound. That's going to cause this B cell to undergo clonal selection. So it's going to divide and divide and divide and divide and divide and create an army of cells that have the exact same dark blue receptor as the first one that was activated. Some of these clones are going to become plasma cells, and the plasma cells are going to secrete antibodies at the rate of about 2,000 per second. And these antibodies are going to molecularly be the same as the B cell receptor. So this antibody is going to be able to grab on to these little orange antigens. Some of our clones are going to become memory cells. And so the memory cells aren't going to fight the infection that time, but a month, a year, 10 years down the road, if you're exposed to the same antigen, then you have these memory cells, and these memory cells are much faster at maturing. And so the memory cells will produce tons of plasma cells. The plasma cells will churn out tons of antibodies. And so you tend to be able to fight off the infection much faster without ever experiencing any of the symptoms of that particular disease. And in that second infection, you're going to get some memory cells that are produced there too. So if you encounter it a third time, you'll get some memory cells for the fourth time, because there's no limit to the number of times we can be exposed to an antigen in our environment. So in primary response, let's say on day 0 is the day that you pick up this virus, right? You're walking through Target, you grab a basket, you accidentally rub your face, and the virus gets in through your mucous membranes. It's going to take about a week for us to go through that stage where we find the appropriate B cell, we activate it, it undergoes clonal selection, and then the clones are going to differentiate into our plasma cells. So around day 7, you're going to start to see your antibody levels start to go up, and it's going to peak right around day 10 or 11, and then we're going to have a sharp fall down of the amount of antibodies. Now, if you encounter that same antigen again, this can be weeks or years or months. If you encounter the same antigen a second time, instead of having a 1-week delay, it's like a 1-day delay. Those memory cells are going to kick into action and start churning out antibodies. So we get much more antibodies. They're much more effective at binding to the antigen, and their levels stay higher for weeks, not just a couple of days. They stay super high for weeks. So while we're looking at our humoral immunity, there are 2 different types of humoral immunity. You have active humoral immunity and you have passive humoral immunity. In active immunity, your B cells are going to be challenged directly by an antigen. Like, you're going to get a dose of the antigen, right? And your immune system is going to say, oh, I recognize that as foreign. We need to get rid of it. So in active immunity, you're going to have a B cell challenge, but there's 2 different ways that you can get that B cell challenge. Naturally acquired is just when you pick up the virus in your environment, like shopping at Target, right? Or grabbing the door handle at the movies, which nobody does anymore because we don't go to the movies. But then there's artificially acquired. And artificially acquired is when we use vaccines. So we trick the immune system into thinking that you have this virus or bacterium by showing it little pieces. Like when you get the COVID vaccine, you're not getting all of the viral proteins. You're just getting like 1 or 2. And it teaches your immune system to recognize it. So if you're actually exposed to COVID, you already have the memory cells. And the memory cells are going to kick in just a matter of hours, 24 to 48 hours, and they'll start churning out those antibodies. So naturally acquired is when you pick it up in the environment. Artificially acquired is when you get a dead or an attenuated. Attenuated means that it's alive, but it's weakened. It really can't infect your cells. You get a little dose of that virus or bacteria, and it just kind of trains your immune system for when you encounter it in real life. And then we have passive immunity, and this is going to be immunity that results from antibodies that are obtained from serum of an immune animal. So your B cells are not directly being challenged. So you've probably heard this in the news lately. They are asking people who are recovering from COVID to donate plasma that has the antibodies in it, right? It's called convalescent plasma, but convalescence is when you are healing. So it's the plasma that has the antibodies in it while you are recovering from your infection. So if you give convalescent plasma to somebody, their B cells are not being directly challenged, but they're using somebody else's antibodies to fight the infection. So we can do this not just by using convalescent plasma from other people, we can actually use antibodies from other animals. So like antivenom that is produced against the venom of like snakes and spiders, they get that by injecting a little bit of the venom into an animal, like a donkey or a cow. The animal's immune system makes antibodies against that venom, and then they take some of the blood from the cow and they purify it, right? And then what you're left with is just like concentrated antibodies. So they're just borrowed. And antibodies don't exist forever. They're going to naturally degrade over a matter of a couple of weeks. So it's a temporary patch to kind of get you over the rough spots. This type of immunity is also conveyed between mother and fetus through the placenta, and it's also conveyed from mother to newborn through breast milk. So her body makes antibodies, and then she passes those to her offspring. And that's one of the reasons why they think that, you know, we've been successful as animals is because when we have our little babies, you know, we kiss all over them, like kissing all over their faces and their little hands and everything. And you're ingesting the pathogens that are on their skin. And so your immune system makes antibodies against those microbes, and then you pass the antibodies to your babies when you breastfeed them. So it's kind of like this whole cycle that occurs. So you can have this conveyed from mother to offspring, or it can be conveyed artificially by giving somebody a dose of what we call gamma globulin or immune serum, just highly concentrated antibodies from another individual or another animal. So we have immunity. Immunity is subdivided into naturally acquired and artificially acquired. Under naturally acquired, we have our active, naturally acquired active, and that's when you just contact the pathogen in your environment. Or we have passive, which is when mother passes antibodies to her offspring. Under artificially acquired, we have active because we have a B cell challenge, and that's when we are going to administer a vaccine. And we have artificially acquired passive, which is when you give an injection of gamma globulin. So let's look a little bit more at these antibodies before we move on to our T cells and our cell-mediated immunity. Now, antibodies are also called immunoglobulins. They are abbreviated with an uppercase I, lowercase g. And these are going to be little soluble proteins that are secreted by an activated B cell, or what we call a plasma cell, in response to an antigen. And antibodies are capable of binding specifically with a particular antigen, right? Each antibody can only respond to one antigen. So this is some computer graphics that show us kind of the shape of what an antibody looks like. Antibodies usually either look like a letter, capital letter T or a capital letter Y, where they have these arms that can grab onto things. Now, antibodies are going to have, there are going to be 5 different classes of antibodies, depending on the genetic makeup of like the stem of the letter Y or the letter T, right? So whatever the amino acid sequences in the stem gives us our 5 different classes. Now, these 5 different classes of antibodies are going to spell out the word MAG. So we have immunoglobulin M, immunoglobulin A, immunoglobulin D, immunoglobulin G, and immunoglobulin E. Immunoglobulin M is going to be found attached to a B cell, so it can function as a B cell receptor, or it can be found free in the plasma. And when it's found in the plasma, it's one of those components that can fix complement, kind of start that complement cascade reaction we talked about. We have immunoglobulin A, and immunoglobulin A is found mainly in mucus and other secretions that bathe body surfaces. So saliva, tears, intestinal juice, and mother's milk. It's also called secretory IgA because it is found in a lot of our body secretions. So since it's found in body secretions, it plays a major role in helping to prevent the entry of pathogens into the body. Immunoglobulin D is also going to be a B cell receptor, and so it's important in the activation of B cells. Immunoglobulin G is the most abundant antibody in plasma. It's the main antibody of both our primary response, the first time we encounter an antigen, and the secondary response, if we encounter an antigen a second, third, or fourth time. This is the only antibody that can cross the placenta, right? So it can cross from mother to fetus. And it's going to function by fixing complement. It starts that complement cascade reaction. And then we have immunoglobulin E. And immunoglobulin E is going to be secreted by plasma cells in the skin, the mucous membranes of your GI tract and your respiratory tract. It's also secreted by cells that are in the tonsils. And our immunoglobulin E plays a really important role in allergies. So if you have hay fever allergies or pollen allergies, or if you're allergic to peanut proteins, this immunoglobulin E is a major player in that process. So antibodies are going to not eliminate antigen from the body, but they are going to inactivate antigens in a number of different ways. So they don't actually like get them out of the body, not directly, but it kind of tags them for destruction. So there are 4 different ways that antibodies can help to tag antigens in the body. And the 4 different ways are going to spell the word PLAN, P-L-A-N. And I should have put them in order in the slide, but I'll read through them in the PLAN order. So the P stands for precipitation. So this is when our antibodies are going to grab on to individual antigens and kind of cross-link them so that they form these large antibody-antigen complexes that are molecularly much larger, and they tend to settle or precipitate out of solution. The L stands for lysis. Now, with lysis, what happens is the antibodies are going to align on the surface of a cellular pathogen, like a bacterium, and it's going to activate complement. And one of the things that complement can do is it can form these little rings of proteins in the plasma membrane of a cellular antigen, which allows water to rush in and causes the cell to burst. So that's another thing that antibodies can do. The next thing they can do is called agglutination. The A in PLAN is for agglutination. This causes clumping of foreign cells that is induced by the cross-linking of our antigen-antibody complexes. So it's similar to precipitation, but agglutination, instead of like grabbing little tiny soluble foreign proteins, it grabs onto like whole entire cells. So when we looked at when you mismatch blood typing and we get agglutination or clumping of the blood, that's because it's the antibodies that are grabbing on. And then the last one is neutralization. That's the N in the word plan. This is when antibodies bind to specific regions on viruses or bacterial exotoxins that block their ability to bind to tissue cells, so they can't damage tissue cells any longer. All right, so this is our PLAN. So when we get antibodies in the plasma that join together with antigens, we get antigen-antibody complexes. So we have four choices, PLAN. You can get precipitation, when small soluble molecules are kind of linked together. They form these large lattices that tend to settle out, and then a macrophage can come along and kind of hoover up the whole thing. L is for lysis, and lysis occurs through complement fixation. The antibodies line up on the surface of a cellular antigen, and they form a complement binding site. And so the complement is then going to form this little ring of proteins in the plasma membrane that allows for water to rush in, and it causes the cell to burst. Then we have agglutination, and agglutination is clumping like precipitation, but it's clumping of cell-bound antigens, antigens that are found on the surface of a foreign cell. And then we have neutralization, and neutralization is going to bind to specific regions of a virus or a bacterial exotoxin, and it prevents their ability to get into our cells, into host cells. Okay, so that's it for humoral immunity. Humoral immunity is provided by the B cells through the production of antibodies. And antibodies are only good at detecting antigens in the body fluids, just like floating by. Now, if things get a little bit further and these foreign substances get into our cells and they're hiding out, the antibodies in the B cells can't see them anymore, right? They're inside of a cell. So B cells are just like, I don't see it. Antibodies can float by and they're like, I can't get it. It's inside the cell. You know, I'm outside the cell. It's inside the cell. So B cells are completely ineffective once the antigen has gotten into a normal body cell. So T cells are going to play that cell-mediated role. They are going to look for normal body cells that are displaying abnormal proteins because they've been hijacked. So a T cell is the opposite of a B cell. Like if it has something that's just like floating by, it's going to be like, so, doesn't bother me at all. A T cell can only see an antigen that has gotten into a normal body cell, and the body cell is acting abnormally, right? It's not behaving. And so it displays an abnormal looking protein on its surface, and the T cell hones in right on it. It's like, oh, yeah, that doesn't look good. I think you're infected. And then it'll go and kill it. Remember, our T cells, when they are undergoing their education in the thymus, they are going to have their antigens presented to them by a macrophage, right? It's not going to see it in its pure, unchopped up form. It's only going to recognize it in its chopped up form. Now, in order to activate a T cell, a macrophage has to grab onto an antigen, engulf it, chop it up, and display a little piece of that antigen on the external surface and show it to the T cell. So that step is called antigen presentation. It's the first step that's required for the activation of our T cells so that they can undergo clonal selection and create an army of clones. So here we are seeing our antigen presentation. So here's our macrophage. It has engulfed an antigen. It brings it into its cell, and then it uses enzymes in the lysosome to chop it up into little pieces, and then it displays a little piece of that antigen on a normal self-protein. So like the self-protein is like the hot dog bun, and the antigen that slides inside of it is going to be the hot dog, right? So the T cell is displaying an abnormal hot dog in a normal hot dog bun. And so the T cell will look at that and go, okay, well, I recognize the hot dog bun, but I don't recognize this hot dog. That's not right. And so it'll attack it. So we have a couple of different types of T cells. We have cytotoxic T cells, and they are going to specialize in killing cells that have been infected with viruses, intracellular bacteria, if they've become cancerous, or if they are tissues that have been grafted from another person. Sorry, still a little choked up. Then we have helper T cells, and helper T cells are like the wedding planner. They have to coordinate and manage all of the other cells of the immune system. And once they are activated, they are going to circulate through the body, recruiting other immune cells to the fight. So they're like the wedding planners. All right, so when a cytotoxic T cell is activated, it's going to go around and it's basically going to take a look at all of the cells in your body, right? If your cells are displaying normal hot dogs and normal hot dog buns, it's going to ignore it. If the cell has become infected with a virus, or if it's become cancerous, it's going to display hot dogs and the hot dog buns, but the hot dogs aren't going to be the right hot dogs, right? It likes Hebrew National and you're giving it Oscar Mayer hot dogs. And it's just like, nope, not doing it. I like Hebrew National hot dogs. And so when it sees that abnormal protein, it knows there's something going on inside that's not right. And so what it does, it can kill that abnormal cell. And the way that they do it is differently from the way that antibodies work. Cytotoxic T cells are going to do something called, they're going to release what's called the lethal hit. And what it does is it has these little cytoplasmic granules, and the granules are going to contain two different types of proteins. Some of them are called perforins, so they form these little pores in the plasma membrane of the cell that's infected. And they have proteins called granzymes. So the perforins create little pores, and then the granzymes are going to move through the pores, and they are going to kill the cell by disrupting the metabolism of the cell. So our cytotoxic T cell is going to bind to this target cell that is displaying an abnormal protein in its hot dog bun. It's going to release the perforins and the granzymes. The perforins are going to form little pores in the plasma membrane that allow for the granzymes to get in. And then the granzymes enter the target cell and degrade the cellular contents. And then the cytotoxic T cell can detach, and it can do this over and over and over again. So it's not like one T cell to one abnormally acting cell. The T cell can kill, and then it can move on and kill again and again and again and again. So they're like the snipers. And then we have a third class of T cell that are called regulatory T cells. When I first started teaching, they used to call them suppressor T cells. And so regulatory T cells are going to release chemicals that suppress the activity of both T cells and B cells once the threat has been eliminated, right? Once we've gotten rid of whatever that antigen is. So they play a vital role in kind of dampening down our immune response once the threat is over. We don't want these activated B cells and T cells to wander around indefinitely, right? They might start killing or attacking normal tissues. So once the threat is over, we want them to go away. They can just kind of like die, a natural cell death. And we have our memory cells in case we ever see that antigen again. So it's no big deal. Now, when we look at our clones, most of our activated T cells in a particular immune response are going to be dead within a couple of days. But just like our B cells, we are going to get memory T cells, and they are going to remain behind for your entire life. And they provide our immunological memory for each antigen that we've encountered over the course of our lifetimes. So even an antigen that you encountered when you were 4, you still have memory cells against that antigen. So if you encounter it again, you're going to get that rapid second response to that antigen. So now we're going to briefly look at organ transplants and rejection, because organ transplants are highly affected by both B cells and T cells. We have 4 different types of tissue grafts that can be done. There are autografts, and autografts are when you take tissue from one individual and move it to a different spot in the same individual. So if you get 3rd-degree burn on your arm, you can take skin from your leg and move it up to the arm, right? So that's an auto. Auto means self. Then there are isografts, and isografts are going to be when tissues are grafted between identical twins. Identical twins have the same DNA, so you should be able to transplant tissues relatively easily without any chance of rejection. Then there are allografts, and allografts are the most common. That is when tissue is taken from an unrelated person. And there are xenografts, and xenografts are when tissues are taken from a different animal species. And xenografts are almost never successful. They've had some limited success with heart valves. So if you have a defective heart valve, sometimes they can replace it with either a pig or a cow heart valve. And I think part of that reason is because connective tissues are not really, they're not supercellular, they're mostly fibers. And so the fibers aren't going to have those genetic markers on their surface the way that a cell would. So remember, we looked at dense, regular connective tissue underneath the microscope. We had lots and lots and lots and lots of collagen fibers, and just a row here or there of those fibroblasts. The fibers aren't going to stimulate the immune system, but the cells will. And so there's some limited success with connective tissues, like the connective tissue that makes the heart valves. Now, autografts and isografts are ideal donors. They are just about always successful given that there's an adequate blood supply and there's no infection. Xenografts are almost never successful, but like I said, they have been able to use pig and cow heart valves. The graft type that's most often used is an allograft taken from a recently deceased person. So if you get a liver transplant or you get a kidney transplant, that's generally going to be from somebody that's recently deceased. Before an allograft is attempted, we have to match up the ABO and other blood group antigens between our donor and our recipient. And we're going to try and get as close of a match as possible in order to reduce the chances of rejection. You need to have at least a 75% match for the transplant to even be attempted. Regardless, after surgery, somebody who receives an allograft has to stay on immunosuppressive drugs for their entire life, because the cells of that tissue are never going to be your own cells, right? They're always going to be somebody else's cells that have somebody else's DNA that produce somebody else's proteins. And so your body is never going to go, oh, okay, it's been here a long time, I'm good with that now. No, it never gets used to it. You have to stay on immunosuppressive drugs forever. Let's quickly look at some disorders of immunity. The first one of these is going to be allergies. Allergies are also called hypersensitivities. This is when you get an abnormally vigorous immune response to something that is just perceived as a threat, but really isn't a threat. Oh, that word should say threat, not treat. It's not a treat, it's a threat. So things like, plant pollen. You're not going to die from plant pollen, but it causes this vigorous immune response, right? So it's perceived as a threat, even though it's not really a threat. And so instead of having a protective immune response, it's actually a harmful immune response, and it damages your own tissues. So an allergen is an antigen that causes allergies. People rarely die from allergies, but you're just usually miserable when you have them. Like this time of year, my pine trees in the backyard are pollinating. I mean, you can see like that pollen everywhere. We also have immediate hypersensitivities, and this is the most common type. They're also called acute hypersensitivities. And remember we said allergies, immunoglobulin E plays an important role in our allergies. So these immediate hypersensitivities are going to be triggered by the release of histamine when immunoglobulin E binds to a type of white blood cell called a mast cell. And mast cells have lots and lots and lots of little granules that contain histamine. That histamine is going to be released. And histamine is a vasodilator. That means it causes your blood vessels to enlarge. They become leaky. More of the fluid leaks out, and that's what makes your eyes all runny. It makes your nose run. You get all itchy. You might get hives. Now, there are severe cases of immediate hypersensitivities, and one of those is called anaphylactic shock. This is going to be body-wide, or what we call systemic, acute allergic response. It is relatively rare, but it can occur whenever an allergen directly enters the blood and circulates through the body. So people who are allergic like to bee stings or peanuts or other types of tree nuts, some people are allergic to penicillin. If it gets into the bloodstream and it circulates, you'll have this entire system-wide response where all of your blood vessels dilate at the same time, your blood pressure will plummet, and that can actually cause cardiac arrest. So it can be deadly. So here we have an allergen, and when we are exposed to an allergen, our B cells are going to become sensitized, and they will start to make antibodies against this allergen. Well, those antibodies, that immunoglobulin E, is going to attach to mast cells. So if you encounter that same antigen again later on, now you have these sensitized mast cells that are going to just kind of flood your body with histamine, which gives us our leaky capillaries. The mucus secreting cells of your respiratory tract become overactive, and the bronchioles that supply air to the little air sacs of your lungs, those are going to constrict. And so that can definitely cause death when it is severe. We also have delayed hypersensitivities, and these are going to be mediated by our delayed sensitivity T cells and by cytotoxic T cells. They take much longer to appear than any of our acute reactions that are produced by antibodies. Instead of histamine, the chemicals that mediate these reactions are going to be released by the activated T cells. The most familiar example of a delayed hypersensitivity is allergic contact dermatitis. So if you are allergic to poison ivy or you're allergic to certain metals in jewelry, after like a day or so of being exposed, you're going to break out in a rash. So it takes a little bit longer. Another type of disorder of the immune system is called immunodeficiencies. And immunodeficiencies can be congenital, that means that you are born with them, or they can be acquired over the span of your life. But both of these are going to be whenever there is abnormal functioning of immune cells, or if the complement does not function properly. The most devastating congenital disease is called severe combined immunodeficiency disease. It's abbreviated SCID, and this is when there is a marked deficit in both B and T cells. So this is like the boy in the plastic bubble disorder, where children are born with virtually no immune system at all, and so they have to live inside of these sanitized, sterile bubbles. Me being a good bit older than you guys, there was a movie in the early 80s, and John Travolta was in it. It was called The Boy in the Plastic Bubble, but probably more related to like your age group. A while back, Jake Gyllenhaal made a movie, and it was called Bubble Boy. It was kind of the same premise, where without the B and T cells, you can't fight off any infections, and so you would die from any type of opportunistic infection. The SCID can be treated in some cases with a bone marrow transplant, but it's not 100% effective. The only hope for survival is to keep all of our infectious agents out, and so these people have to live in these sterile plastic bubbles. Another immunodeficiency is AIDS, acquired immune deficiency syndrome. So this one is not congenital. You're not born with it. It's not coded for in your DNA, but you can acquire it during your lifespan. It is the most important and most debilitating acquired immunodeficiency, and it disables your immune system by interfering with the activity of the helper T cells. So remember, we talked about the helper T cells, kind of like the wedding coordinator, and make sure, like, the flowers and the band and the food and the music and, everything is taken care of. Well, when you don't have a helper T cell to help, everything just falls apart, right? There's basically no immune response at all if you don't have any helper T cells. And then we have autoimmune diseases. And so this is when the immune system can no longer distinguish self, like your normal self proteins that are in the hot dog buns, versus non-self. And your immune system starts to produce antibodies and sensitize T cells against your own tissues. And there's a couple of different common types of autoimmune diseases. Multiple sclerosis is one that most people have heard of. That's when your immune system attacks the myelin sheath in the brain and the spinal cord. So it kind of short circuits those action potentials, those electrical signals. There's myasthenia gravis, and that's when the immune system attacks the neuromuscular junction. So it impairs the ability of nerves to talk to skeletal muscles. And then there's Graves' disease. And Graves' disease is when the immune system attacks cells of the thyroid gland and you get too much thyroxine. Other autoimmune diseases, we have juvenile diabetes mellitus, the one that they call type 1 diabetes. This is when the immune system is going to destroy the beta cells of the pancreas that secrete insulin. There is systemic lupus erythematosus, or SLE. This occurs mainly in females, and it particularly affects the kidneys, the heart, the lungs, and the skin. There is glomerulonephritis, which is going to be severe impairment of kidney function because the immune system is attacking the kidney tubules. And then there's rheumatoid arthritis. And rheumatoid arthritis is when the immune system kind of systematically attacks the tissues that are in the joints. Reasons why autoimmune disorders actually exist, because you would think with suppressor T cells that the suppressor T cells or the regulatory T cells would kind of dampen things. There's a couple of different theories, even though I don't think any one thing has been highly endorsed by the medical community. One thought is that there is ineffective programming of our lymphocytes. So self-reactive T cells and B cells are not eliminated by the thymus and the bone marrow. Remember I said the thymus is going to eliminate about 98% of the T cells that go there to be educated. It's a very expensive process. Well, maybe some of those escape, right? Maybe they actually escape and get into circulation. Another thought is that there might be the appearance of self-proteins in circulation that have not been exposed to the immune system when your immune system was at its height, which is during childhood. So that new self-antigen may appear as a result of maybe a mutation in the DNA of a tissue cell, or it could be an alteration of the self-proteins. And then the last theory on autoimmune diseases is cross reactions of antibodies that are produced against foreign agents with self-antigens. So the antigens are produced for a foreign antigen, but some of your self-antigens might look similar, and so those antibodies could attack.